Welcome to the Garnet Presents webinar. So the eagle-eyed amongst you will see that both Joe and myself are wearing something different today, because this is actually a recording the day after the live webinar went out. We had some issues with the sound that it didn't record. So Joe is going to kindly repeat his webinar now, so we can get to hear about the information that he gave out. So Joe's joined us. Joe McKenna is joining us from Oxford Brookes University. He does PhD at Imperial College before moving over to Oxford Brookes to work, and he really works in the imaging centre that they have there, and they do some excellent work off the back of leadership of Chris Hawes and also John Runyon's there. So thanks very much, Joe, for joining us again today, and we look forward to hearing what you have to say.
Thanks very much, Geraint. So first of all, before I start, I'd just like to say thank you to Geraint for giving me the opportunity to talk and for organising this seminar series. I think it's been a really good series showing the breadth and the strength of plant science in the UK, and the calibre of talks have just been exceptional. So hopefully I won't lower that bar too much with the presentation I'm about to give you guys. So yeah, my name's Joe. I'm an independent research fellow at Oxford Brookes, and I work on a number of different things, but what I'm going to talk to you about today is the plasma membrane. And this work was started when I was a postdoc in John Runyon's group at Oxford Brookes, and John was really at the heart of a lot of this work. And then now that I'm starting to step into my research independence, I'm taking some aspects of it forwards, or trying to take some aspects of it forwards on my own. And what a brilliant time to try and become research independent during the pandemic. So thank you all very much for your time and to listen. So I've always really been interested in how you get structures forming in cells, how you organize things, and how you organize things in a way that allows the cell to function. So I'm very interested in actin cytoskeleton, how that's organized, the nucleus, how that's organized. What I'm going to talk to you about today is work that I'm doing on plasma membrane and its organization. And the plasma membrane really is fundamental for most processes that happen within a plant. There's obviously the textbook view of it separating the inside from the outside, but in addition to that, it's full of proteins, and these proteins in the plasma membrane, they do lots and lots of different functions. So they can do things such as they can produce the cell wall, the extracellular matrix, they can interact with the cell wall and nuclear actin within them, they can are involved in hormone perception, hormone transport, so morphogenesis, absolutely fundamental for. They can perceive stresses from the environment, so through receptor kinases such as FLS2, EFR, these kind of things. So they're really fundamental for the cell functioning in a normal state and adapting to responses in the environment. And when we think of the plasma membrane, we really should think of it more as a continuum. So what I mean by that is you've got the plasma membrane, but then you've got it being pressured, pushed, really with great turgor pressure, pushing it into the cell wall. Now, EM measurements have said that there's about a 10 nanometer gap between the plasma membrane and the cell wall. So you think about all these proteins with extracellular domains, they're really being pushed into this extracellular matrix. You've got the cell wall, you've got the plasma membrane, and then underlying that, you've got the cortical actin and microtubule cytoskeletons. And these are really important for many different roles as well. And what's really interesting is that some of these domains, some of these elements of the continuum can interact with each other. So it's known that cellulose synthases, they move through the plasma membrane, they push cellulose out into the cell wall, so the plasma membrane regulating the cell wall. These cellulose synthase, their movement is regulated mostly via microtubules. So microtubule, they're moving along, being guided by the microtubules. So it's effects there. Actin, the formins are known to be anchored into the cell wall, pass through the plasma membrane, and can nucleate and interact with actin in the cytosol. So there's a number of different elements to this continuum. And when we were designing our experiments, most of the hypotheses that we were trying to draw together, we're looking at how we can separate these different functions out, how we can tell whether these are interacting with each other or how they're regulating each other. And the plasma membrane it used to be seen as kind of a sea of molecules. So the fluid mosaic model suggests that there was, it's just a random sea of molecules floating around and bumping into each other and moving all over the place. But that's really not the case. So lots of work from mammalian cells and plant cells over the past 10 years, even longer, has come up to the idea now that actually there's structure within the plasma membrane. There's protein heterogeneity. So there's islands of proteins, and how these islands are formed, how these structures are formed, are thought to be really, really important for their function. And there's a fantastic paper from Thomas Ott and his group in PNAS in 2018, and they showed really nicely there that for an infection thread formation during nitrogen symbiosis, one of these marker domains, the remorin, was fundamental for that. So we know that membrane nanodomain, that a lot of proteins are in membrane nanodomains. And what can regulate this organization? So before I go on to that, I want to talk a little bit first about the work which had gone on in the lab previously, which kind of guided us through where we might want to start looking. And all this work was done in John's lab by John's previous postdoc, Alex Martinier. Alex is a rock star of plant cell biology. His work is just absolutely fantastic, and this was by no means any exception. So what Alex did was, Alex really nicely showed that when you do a technique called FRAP, so fluorescence recovery after photobleaching, what happens is you bleach an area of the membrane and you then image over time the recovery of that fluorescence into that area. And this gives you an idea of the lateral mobility of bulk protein movement, so lots and lots of proteins moving. And as you can see here, what was really nicely found was that all these proteins seem to be recovering at slightly different rates in the membrane. So this was an interesting observation. And it's been known for a while as well that in the plant plasma membrane, things move an order of magnitude slower than they do in animal systems. So this really got Alex and John thinking, well, what can be involved in this? And they did a really elegant series of experiments where they took marker lines and they protoplasted them. So they digested and they removed the cell wall. And after they'd removed the cell wall, they then imaged recovery. So you can see here, this is immediately after protoplasting here. This is just before the bleach has occurred. And what happens is when you bleach it, you get a relatively rapid recovery after 100 seconds. Now, when you wait, and you wait 48 hours and you let the cell wall recover, what you see now is the pre-bleach. And then when you bleach this protoplast, you can see the recovery hasn't gone as quickly. So this implies that the cell wall in this minimal membrane, the cell wall has been very important in regulating its lateral mobility, how quickly it can move in that domain. And he backed this up with plasmolysis as well. So plasmolysis, where you separate the plasma membrane from the cell wall. And he showed really nicely here. So in this control example, you bleach and recovery doesn't happen very quickly. And if you plasmolyze the cell, if you separate the plasma membrane from the cell wall, you can see really, really nicely here that recovery happens quite a bit quicker. So building on from this work, what we then wanted to do was we wanted to look with slightly higher resolution and try and see if we could image these membrane nanodomains. Now we used a technique that I'm going to go on to in a minute. Lots of other groups have imaged membrane nanodomains in plants. We were by no means the first, but we took a slightly different tact because we used a technique called AereScan super resolution. So the way a normal confocal works is you illuminate your sample, you collect the emission, it passes through a number of beam splitters and filters. It's then all the out of focus light is blocked by a pinhole and it goes into a detector. And this is how you generate your image. And here, what you can see, so this is with PIP2A and with BRIA1, you can see these are membrane sheets in hypocotyl cells. And you can see quite nicely here that there's a lot of noise. You can maybe make out areas where there is something going on, but there's a lot of noise there in the sample. So then when we tried the AereScan technique, so AereScan is a technique developed by Zeiss, and the way that this works is instead of the outer focus light being blocked by the pinhole, you have some zoom optics here, and what these do is these spread all of the light across this specialized detector. What this specialized detector does is it allows us to, it's actually 32 little detectors all combined together, and we can take data from each one of these detectors and subtract them from each other. What this does is it allows us to reduce the point spread function, which allows us to get slightly higher resolution. So it's a really, really good way of doing super resolution, but it means that you can do it on your samples exactly as they were with all the normal fluorophores that you were using, and you don't need to change anything at all. You just click a button on the confocal and instead of using normal confocal, you use Aeroscan. So we found it very useful. And when we look now at these images, what we can see really nicely is you can start to see structure here. You can start to see these nanodomains within the plasma membrane in both these conditions. And what I would say is what you're seeing here is the exact same cell, the exact same area imaged one after the other, once initially with confocal and then with Aeroscan, with the exact same laser power and the exact same detector settings, although obviously going through a different detector. So what Aeroscan allows us to do is it allows us to resolve these protein nanodomains in the plant plasma membrane, and it gives us a spatial resolution limit of 120 nanometers. So this means that we can spatially see these nanodomains. This is the tool that we use to be able to go and to characterize them. So we then went on to look at some biologically some different proteins in the plasma membrane bound, and these proteins, which I'm going to talk about a lot during this talk, are the pathogen receptor PAMP receptor FLS2 and the hormone transporter PIN3. And we focused on these two because they both have extracellular domains and they function in completely different pathways. So FLS2 we didn't treat with the ligand flag22, so it should always be inactive, whereas PIN3 there will be some auxin present during the normal development of this plant, so it might be active. And we reasoned that if we found something which affected the nanodomain characteristics of both of these proteins, it's probably quite a good indicator that it has a general role in the formation of nanodomains. And what we did is we did our Aeroscan imaging, like you can see here. So here's the view that we saw through the confocal. Here's a magnified view. And we then drew lots of line profiles over these and determined a measurement called the full width half maximum. And this is the measurement of detecting how big something is from microscope data. So we did this for both FLS2 and PIN3. And we found really interestingly that what you can see here is both of these form nanodomains below the resolution limit normally for GFP, which is 250 nanometers. They form nanodomains below that, but also the nanodomains appear to be slightly different sizes. So PIN3 is slightly bigger than FLS2, and this was quite interesting. We also included the Ramorin 1,3 as a control because Ramorins have been shown to form nanodomains that you can see above the resolution limit. You can see with normal confocal microscopy. And if protein nanodomains is piquing any interest in your head at all, I really highly recommend you go and look at Iris Shark and Thomas Ott's paper from 2014 in Plant Cell. It's a real tour de force in characterizing Ramorins and the Ramorin family. And they imaged every member of this family, and they showed where it was throughout, how the nanodomains looked and how they structured. It was really, really fantastic paper. But one thing that I really want to stress from that paper is they showed really, really nicely that depending on the different tissue type, it's one of the later down figures, depending on the tissue type that you image the Ramorins in, the nanodomain structures might look slightly different. So to stress, all of the work I'm showing you, we did in the hypocotyls of 5-day-old Arabidopsis seedlings. So if you're doing Aeroscan and you're looking at these proteins and you can't see this, but you're looking in different tissue, that might be the reason why. So all of the work I'm going to present to you was done in 5-day-old Arabidopsis hypocotyls. We also showed that, although I'm not showing for the sake of brevity, I'm not showing the data, that BRI1 and PIP2A also have differently sized nanodomains that were below the resolution limit of normal confocal microscopy, but we were able to resolve with the Aeroscan system. So this is really interesting. So Alex showed previously that the lateral mobility, when you look at bulk protein movement via FRAP, is different for proteins. But now what we've gone and done is shown with Aeroscan, our tool for looking at spatial resolution, how small these nanodomains are. We've shown really, really nicely that the proteins we've looked at seem to occupy or exist in differently sized nanodomains, which is really, really quite interesting. And I should say nanodomain, I should have said this in the introduction, it's defined as something from about 20 nanometers, usually up to about 300, but anything less than a micron. and we use that term nanodomain to describe these protein heterogeneities within the plasma membrane. So Airiscan was a fantastic tool to allow us to get the best spatial resolution we needed to be able to quantify these nanodomains and to be able to look at the structure of these nanodomains. But it's too slow, or it was too slow, to allow us to quantify single particle movement over time. So these domains can move relatively quickly. So we were unable to do that at the time with Airiscan. As time's moved on, now we have Airiscan techniques where we actually can track them over time. But when we were doing this work, we weren't able to do that. So we had the spatial resolution with Airiscan, but we couldn't track them over time. So what we did is we turned to a different technique, which gave us the temporal resolution. So being able to see very fluidly the movement of single particles over time in the plasma membrane. And that technique that we turn to is called total internal reflection fluorescence. Now, I'm a massive microscope geek, so I love microscope techniques and everything. This is the last one I'm going to introduce you guys to, I promise, and then we'll be biology all the way after this. So what TURF is it's a clever way of being able to cheat your way around being able to image things quite quickly. So instead of with a confocal where you shine the laser straight through the sample, the way TURF works is you shine the laser at a critical angle when it hits the cover slip. This causes the vast majority of the energy, laser energy, to be bounced back into the system and to basically go away. But a little bit of the energy generates something called an evanescent field, which passes about 100 to 200 nanometers through the sample. And this passing about 100 to 200 nanometers through the sample excites only things in the plasma, only fluorescent proteins in the plasma membrane, or very cortical things such as the actin cytoskeleton or ER. So you can beautifully see here the kind of temporal resolution you can get with the ER and the actin cytoskeleton. So it's not necessarily a super resolution technique, so to say with XY resolution, but it does allow very, very, very quick imaging in a very short Z depth. So this is one of the reasons why we use this technique, because it allows us to very, very rapidly image samples. So some of the movies that I'm going to show you, we were imaging at up to 100, 200 frames per second. So very, very, very quick imaging. So how do we, it's very nice to be able to acquire this data, but how did we go along and analyze it? And all of the credit for the analysis has to be given to Dan Rolfe and the team, so Stephen Webb and the rest of the colleagues at STFC, so the Science and Technology Facilities Council funded Rutherford Appleton Laboratories, the central laser facility. And we collaborated with them, Alex had collaborated with, and John had collaborated with them previously, and we collaborated on them with this work. And these guys are real experts in single particle imaging and single particle analysis. So Dan developed the software, it's a whole pipeline he's got, which allows us to be able to track these proteins and their movement over time. So all credit to Dan and the CLF team for being able to do this. Now, what I'm going to show you is the most breathtaking, mind-blowing, amazing video you guys are ever going to see. Okay, are you ready? There you go. So I will caveat that with, obviously there's an issue at the moment with the quality of, depending on the screen you're looking at, reflection, what have you, of the screen, but hopefully you can see little blinking dots moving through a membrane, flashing, moving and going away, flashing, moving and going away. And what you're seeing here is individual particles in the plasma membranes. This is LTI6B, a marker line, moving through the plasma membranes. This is individual particles, the machinery of life moving through this membrane. And what we can do with the software which Dan and the team developed, we can initially identify every single one of those points over time. And then what we can do is start tracking them over time. So knowing that this molecule has moved here and it's moving here, and this gives us now a really, really good idea of how these molecules are how these particles are moving in the plasma membrane over time. Now, the way we glean data out of these is we produce something called a mean square displacement curve. And what this mean square displacement curve means is it's a way of being able to show graphically exactly how the proteins are moving over time. So if you imagine something which has absolutely no resistance, so let's say a bullet or a rocket fired in space where there's no effect on it, you get a complete linear line, as you see here in yellow, a complete unconstrained curve, mean square displacement curve like this. If you had something which was being actively transported, what you would see was a upwards trajectory like this. However, if something is being constrained, it's being slowed down, constrained in the membrane, what you would see is a lowering down curve, such as the one here. Now, what happens when we look at proteins with a real biological function? So here, what we've got is a number of different proteins. We've got the marker protein LTI6B, we've got PIP2A. These are both driven by 35S overexpression lines with a photoactivatable GFP. We've also then got FLS2, the PAMP receptor, tagged to GFP, and PIN3, the auxin transport protein, tagged to GFP. And hopefully what you can see is these slight differences between these proteins over time. Now, when we go and we look at this data and we quantify what the differences are, we really nicely see that, so here we've got just the tracks drawn in an area over 60 seconds. Here we've got the mean square displacement curves. You can see that the proteins are quite different. And what's really interesting is if you look at the constrained diffusion, you can see here all of the biologically relevant proteins, so PIP2A, FLS2, and PIN3, these all have different lateral mobilities in the membrane at the single particle level. So if you remember, Alex showed bulk movement when you do FRAP, there's differences between the proteins. And when we go now to the single particle level, if you remember with the ARIE scan, we showed two of these proteins exist in differently sized nanodomains. And now we're showing with single particle tracking that the proteins seem to move at different speeds. But where the story gets really interesting is if you now look at the constrained region data down here. So this is what this data is doing, is fitting a model. So we know that we've got constrainment going on here by the shape of the curve. And if we fit a model here, which allows us to predict the constrained region, so this is the area within which these particles are moving, you can see there's no significant difference between the three biologically functioning proteins, PIP2A, FLS2, and PIN3. So they seem to be constrained within a similar region from the data that we're showing you. So then our hypothesis went to, well, what could possibly be regulating the constrained diffusion, the nanodomain size, and this constrained region, which we see here. So initially what we did was we looked at, we went back to our idea of this cell surface continuum, and we messed with the two different components of it that weren't the plasma membrane. So for the sake of brevity, I'm not going to talk much about the actin and microtubule data. Basically, we saw with FLS2, the constrained diffusion was increased when you perturbed either of these cytoskeletons, but the constrained region wasn't, and we saw no statistically significant effect for PIN3. Other people in the literature have gone on and shown really nicely that depending on the protein, sometimes you get an effect with one cytoskeleton or the other, or sometimes no effect at all. So this looks like the cytoskeletons, both the microtubule and the actin, seem to be able to differentially regulate proteins depending on what they are. It seems protein specific. One thing I should also say, sorry, I should have said this right at the beginning of the talk, is I'm not talking about the lipid components of the plasma membrane at all during this talk. They are absolutely fundamental for the structure of nanodomains within the plasma membrane. Absolutely no doubt there. There's amazing work from Yvonne Gellet and Sebastian Mongrand's groups looking at this, and we are, yeah, I'm not mentioning that at all. It's very, very important. We're just, in this work, we really focused on just these two different parts of the continuum. And what we did then was and so there's a nice Ronald Reagan quote for you there, tear down the wall. So we impaired the cell wall. And we did this in two different methods. We used DCB. So what DCB does is it inhibits cellulose synthases. So it stops cellulose from being produced. And we used EGCG. What this does is it inhibits proteins called pectin methyl-esterases, and this causes alterations in the pectin status of the cell wall. So we impaired the cell wall in two different ways, impairing two different structural components of it to see what happens. Now, I'm going to let this video play for a while. What you've got here is we've got, first of all, FLS2 along the top and PIN3 along the bottom. We've got DCB treatment here, and we've got EGCG treatment here. So we're inhibiting cellulose biosynthesis and pectin methylation status. And hopefully, over time, you guys can see that the proteins at the single particle level appear to have when we impair either the cell wall or the EGCG, there appears to be different effects. Now, when we go and we quantify this, if general rule for single particle analysis, if you can see it with the naked eye, it's going to be very significant when you go and do the analysis. When we went and did the analysis, again, for the sake of brevity, I'm only showing you here EPLS2. I'm not showing you pin 3. But when we went and did the analysis here, what you can see beautifully, and it's exactly the same for pin 3 as well, as we reported, with the constrained diffusion is increased. So the proteins are moving faster within the plasma membrane. They're diffusing faster through the membrane. And the constrained region, so if you remember, this was the same for all the 3 biological proteins we looked at, the protein constrained region is increased as well. So the area that they're being constrained to in the membrane is increasing over time as well. So this is really, really interesting. And if you remember, as we've gone through this talk, Alex initially showed that the cell wall was important and proteins, when you looked at bulk mobility, moved at different rates. What we've done is we've shown that proteins seem to exist in differently sized nanodomains, which other groups have shown as well. We were not the first to imply this, but we did with this technique area scan. We've then gone on to show that these nanodomains, the constrained diffusion seems to be different, so they're moving at different speeds within the membrane, but the constrained region seems to be the same. When we mess with actin and microtubule cytoskeleton, there appears to be differential effects. But really interestingly, when we mess with the cell wall, and we're messing with it in 2 very, very different ways here, we can increase both the constrained diffusion and the constrained region. And one thing that's also worth remembering, that these treatments, so an hour of DCB or an hour of EGCG is really not a very long time. Usually for any transcriptional, for the start of transcriptional effects or any kind of compensatory mechanism, so lignin being deposited, for example, you have to wait 4, 7, or 12 hours with DCB or with a similar drug isoxafen. But here we're seeing very, very, very quickly big changes in the diffusion and the constrained region of single particles in these in the plasma membrane. What we then did is we went backwards and we asked, well, when we impair the cell wall in these 2 different ways, if we use area scan imaging, do the nanodomain size change? And as you can see really nicely here, with compared to the 1 hour mark, when we do either DCB or EGCG, the nanodomains increase in size. So we've shown that when you impair the cell wall, you get proteins moving faster, diffusing faster through the membrane. The constrained region they are occupying seems bigger, and the nanodomains are larger in size as well. So we think this really nicely shows that the cell wall has a real fundamental role in regulating the size of nanodomains, in contributing to nanodomain formation and dynamics. Now, what I should say is, in addition to the techniques we've shown here, we've also done enzymatic degradation of the cell wall with a number of different enzyme cocktails. And what this does is this causes the nanodomains to increase in size as well. And we've also done plasmolysis with a single particle tracking. So when you separate the plasma membrane from the cell wall, salt treatment, for example, we also see that the proteins move faster and in larger constrained regions. This really does imply that the cell wall has a really big role in regulating the size and dynamics of these nanodomains. So to summarize briefly, so plant plasma membrane proteins with extracellular domains exist in nanodomains. That's what we've shown. The axin, and this is the same for every single biologically functioning protein which had an extracellular domain that we've ever imaged with area scan imaging, we always see nanodomains, and differently sized nanodomains, as I've said here. Actin and microtubules have an interesting role. They seem to be able to differentially regulate some proteins and not others. But the cell wall, so this extracellular matrix, has a real role in regulating the mobility or the dynamics of these proteins in the plasma membrane. They're the nanodomains in the plasma membrane. And this kind of makes sense because if you remember, I told you plasma membranes being really tightly pushed into the cell wall. And if you impair that over time, it makes sense that there would be changes in the dynamics of proteins in the membrane. So we're really suggesting that the cytoskeleton, but definitely the cell wall, contribute to nanodomain formation and maintenance. So I'm now very quickly going to go over what you definitely shouldn't do during a talk, and I'm going to mention everything wrong with what I've told you. Well, not quite everything wrong with what I've told you, but where I'm planning to go forward with this work. So we've shown really nicely, as I said, that the cell wall has a role in slowing proteins down, basically in regulating the nanodomains. So my question is, what's the point? Why hit the brakes? What effect does this have on the signaling abilities of these proteins? So what's the functional role that this cell wall drag has on these signaling pathways? And if you think about this, this is very, very, very important, because during a normal plant, a crop out in the field, for example, will be going through a number of different stresses in the day, whether it's the moisture conditions, the water conditions, whether there's droughting, whether it's being attacked by pathogens, and all of these can affect this interaction between the plasma membrane and the cell wall. And I've told you that a relatively light, only an hour's treatment of these impairing cellulose biosynthesis or pectin methylases causes these big changes in protein dynamics. So how does that affect downstream signaling? So I'm also really interested in, is there a role for what is this term being coined in the mammalian field, mechanoimmunology? It's been shown really nicely in the mammalian field that, for example, the T cell receptor, it's how tightly the receptor binds its ligands depends on the mechanical properties of the environment it's binding. Is this similar in plants? So the way I'm testing this is I'm looking at receptor ligand binding efficiencies. So can I visualize at the single particle level how efficient receptors are at binding ligands? We're doing this with the reporters mentioned here and fluorescently labeled ligands. I'm also looking at protein complex formation during this role. And if anyone has any feedback on this, I would be absolutely love to hear it and hear anything you've got to say. So on that note, I'd like to say thank you to a few people. So the Oxford Brookes gang, so the whole plant cell biology team, all fantastic. John Runyons, who the work was started in his lab during postdoc with him. Chris Hawes, I'll come to in a minute. Francis told me he was an amazing PhD student at the time. The guys at the STFC Rutherford Appleton laboratories are just brilliant. So Dan Rolfe, who built this whole software analysis system, Stephen Webb, Stan Botchway, and Marissa Martin-Fernandez are the key guys involved in this piece of work. Funding from the BBSRC, the Science and Technology Facilities Council. Just found out I've got my access grant to Hull renewed, which is not renewed, so I've got a new access grant there, so I can carry on this work there really nicely. And thanks to Oxford Brookes, who have funded me as I take steps towards independence. So the only thing I'd like to say to finish is we lost a real titan of cell biology last year. So sadly, with the passing of Chris Hawes, who was just such a wonderful cell biologist. And Chris was all about the science. So the best way that I can acknowledge him is to say, in my opinion, where cell biology is at its best is where with one very well-planned experiment, one very well-planned image or movie or figure, you completely believe a concept. And Chris was a real master of being able to do this. He was able to plan experiments brilliantly, which very nicely demonstrated, proved his concept, his hypothesis. And the two best examples, I think, of Chris doing this are two papers I put here. So Stacks on Tracks and Grab a Golgi. And if you want to know Chris and you want to see amazing cell biology, just check these two papers out. They're really good. So thank you very much. And on that note, I am finished.
Thanks very much, Joe. So super interesting then, and a fitting tribute to Chris there at the end. So let me ask a couple of questions that people asked during the seminar. So one from Stuart Sullivan, who's in Glasgow University. He asked about the cell wall and the effects. So the effects on the nanodomains, whether it's due to the primary or the secondary cell wall.
Okay, yeah. So really, really good question. And I imagine, I don't have the evidence, I imagine it could really affect the dynamics depending on the composition of the secondary cell wall. That being said, all this work was done in 7-day-old Arabidopsis hypocotyl cells. So this would be primary cell wall. Whether Arabidopsis has a secondary cell wall, going back to my PhD days and debating this with the Timber Tech guys, some people might not agree with, but yeah, so it would, I imagine it could affect it, but the question is we don't know. So obviously with the secondary cell walls coming, it would be affecting plasma membrane, and it could be affecting dynamics of proteins differently depending on its composition and depending on the constraint. So if it's much stronger, the secondary cell wall, and you're getting turgid pressure really pushing these proteins in, I could very well imagine you'd see a difference.
Okay. And so second question, this is from Petra Bovink, who was the lead author on the Stacks on Tracks paper that you mentioned, or the first author on that. So she asks about the key technique about this particle tracking that you show. And she asks whether there's any work on verifying that from image to image that the dots that are identified are exactly the same dots, or is there a possibility that one flashes out of existence and is picking up another one?
Yeah, so it's a really good question, and I will try and answer it as best I can, but I will also tweet the original paper about the image analysis method. So the beauty of turf imaging is we can image really, really quickly. So the problem with the area scan imaging was if we tried to do this kind of nanodomain, you'd see a dot here and then it would be here and then it would be here. And how do you know that this dot definitely was, can't be done because we didn't have the temporal resolution. Turf allows this. You can image really, 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 really quickly, which allows you to have this temporal resolution. What the image analysis does is it looks at lots and lots and lots of different criteria. So is something the right size to think it's a single particle? Single molecule? Is it the right size? Does it have the right blinking characteristics? Is it bleaching? So fluorescent proteins bleach in very defined ways. Is it doing this? So what this does is this allows them to build, they've built the software so that it takes all of these aspects into respect. And for example, if this protein here is now over here and it's 4 times brighter, it's unlikely to be the same protein, the same particle. So it takes all of this into effect over time. That being said, is there a chance that the molecule here is now here and there were very, very, maybe, but we were tracking thousands and thousands and thousands of molecules and particles to be able to do this. So, you know, a couple possibly, but I very highly doubt that. But Dan Rolfe and the guys are really the experts, and you know, they built this software and they're very competent in it. It's been used for lots of different things. So in mammalian cells, doing exactly the same thing, but in mammalian cells. So
okay, cool.
Yeah, I think we're okay.
Okay. All right. So I think we'll leave it there. Thanks very much, Joe. Super interesting talk, and thanks for giving it for the Garnet Presents webinar series. Thanks.
Thanks. Bye, guys.